Okay, I want to start with a couple corrections. I've gone back and listened to the first two sessions and realized that, I make mistakes all the time. Some of them are trivial. So, for example, Brecher and Cohn's paper came out in 1969, not 1967. It was, of course, Archimedes and not Socrates who jumped out of the bath and ran around. And some of them are not so trivial. For example, in going through co-stimulators, I said that CD40 ligand was a strong co-stimulator of T cells. And that's actually incorrect. There is one paper which is missing a very important control, which suggests that CD40 ligand is necessary during T cell development and that it signals into T cells. But most of the evidence is that CD40 ligand is used by T cells to bind to CD40 to educate the cell that has CD40. So it's not so much that it's a strong co-stimulator by itself of T cells, it's that it's used by T cells to superactivate or license antigen-presenting cells. So in a way, that also makes them more co-stimulatory for T cells, but it's not the CD40 ligand itself. Right? So that was an important one to fix. Okay, so today is tumors. Last week we did fetuses and surgical transplants, right? And the conclusion there was that even though the fetus is foreign, it's not rejected because it doesn't look dangerous, right? At least if it's doing its normal development. And that surgical transplants are rejected because they look dangerous, because of the damage done by the surgery or the reperfusion injury, or even sometimes the disease that killed off the first graft. So if fetuses aren't rejected because they don't look dangerous and transplants are rejected because they do look dangerous and they're both foreign, why aren't tumors rejected? So we know that there are tumors that express foreign antigens, either virally encoded or mutated proteins or molecules that were up really early in fetal development, long before the immune system arrived, and then get turned off. So early fetal antigens. Or they sometimes express much higher quantities of self-antigens than the normal tissue. So these ought to be rejected, and yet they're not. And when Ephraim first suggested that this was a problem with the self-non-self model, I patted him on the head and said, tumors are immunosuppressive. And there are a lot of people that still say that. There's the myeloid-derived suppressor cells in tumors. There's the T regulatory cells in tumors, and so on. Tumors are immunosuppressive. But we know that the immune system can get rid of tumors. So why doesn't it? So for a while, it was thought that it does get rid of tumors. It gets rid of most of them. The idea when I was a graduate student in the 70s was that we get a tumor every 3 minutes or so, and the immune system gets rid of them. And when we get a tumor, when a tumor actually grows out, it's because the immune system couldn't get rid of it. And that was a theory until about the late 70s when the nude mouse was discovered. Has no T cells, has no thymus. Actually, nude mice have a few T cells from their mothers. Just for those of you who work with nudes or ever plan to work with nudes, be careful. Because nude mice in most colonies are bred from heterozygous nude mothers. And that's because nude mice have no hair, and they're not very good at keeping temperature control, and so they have trouble during pregnancy. And so the way people breed nude mice is they have heterozygous mothers, which have hair and thymuses, and homozygous fathers. And so half the litter is nude and half the litter is hairy. Well, depending on what colony and how they're bred, nude mice have T cells from their mothers. So be careful when you find nude mice. So for a while, people thought nude mice had T cells because they were growing up in the omentum. Well, actually, it's from their mothers. Anyway, nude mice, 50% of them don't have T cells from their mothers, and they don't tend to have a higher incidence of tumors than normal mice. And so the idea that the immune system is constantly surveying, looking for tumors, kind of died. And then the rag mouse and the skid mouse were discovered, and they don't tend to have many more tumors than normal mice either. There's been a resurgence of the idea of immunosurveillance. And there is some evidence that if you take a T-cell deficient mouse and you give it like MCA under the skin, by themselves they don't have more tumors than normal mice, but if you give them tumor-inducing agents, they will get more tumors than normal mice. I don't think that's very good evidence for immunosurveillance, but there you go. It has recrudesced the field. So why do I think that tumors on the whole don't get rejected? So first, we no longer think that you get a tumor every 3 minutes. We think that tumorigenesis is rare because of genes like P53 and so on that monitor cells and tell them to die when they do bad things. And when a tumor does arrive, I don't think the immune system in general is going to respond to it. And the reason it's not in general going to respond to it is that tumors don't look dangerous. You know, what's wrong with a tumor? What's wrong with a tumor is that it's a cell that has lost growth control. So it's dividing when it shouldn't, right? It's making a bunch of cells. It's not looking dangerous, at least in the beginning. It's simply a cell that has lost growth control, right? It's dividing or multiplying. I remember the day that I discovered that division and multiplication in biology are the same. Yeah? And suppose this is a tumor that was expressing tumor antigens in MHC. Let's call them T antigens. And suppose you had a T cell that could see that. This is a viral antigen, a mutated antigen, an early embryonic antigen. What's going to happen to that T cell? Somebody tell me. It'll get signal 1. And?
Not signal 2.
And not signal 2. And? Die. Right? Signal 1. Go ahead.
Hi, Polly. Yes, I was thinking about it, but I also tried to relate that to
Wait. If you're going to talk for a long time, you have to go to the microphone. Right? If it's just a quick question I can repeat, then you can just say it. Yeah?
So I said I was thinking about the foreign antigen and relating that to the peripheral tolerance we talked about last week. So, and you just said like a few minutes ago that tumor will express high amount of self-antigen.
Can do.
Does the dose play a role, antigen dose, play a role in peripheral tolerance?
Good question.
So, because in that way they are still expressing the self-antigen. But some of those self-antigen will activate T cells, even though it's only signal 1. And most tumor will have the inhibitory co-stimulated, as we said. So how come some of the T cells become activated?
Okay, so that's more than one question. So let's do one first. All right, so first, does the dose of antigen matter to tolerance? And there are theoretical reasons for saying yes. So this was originally thought about by Mel Cohn, who I still think is the greatest immunological theoretician out there. And he was talking about tolerance in the thymus. And here's what he said. All right, so first you have T cells of varying affinity, or avidity, since they have more than one receptor. But let's talk about affinity for any particular antigen. At what avidity cutoff do you want to induce tolerance? And he argued that you want the same avidity cutoff that would induce activation in the periphery. If the avidity for tolerance is lower than the avidity necessary for peripheral activation, right, then you're going to tolerize T cells you don't need to tolerize. And T cell development is a pretty complicated thing. It takes energy to do that. So you don't want to kill off a whole bunch of cells that you don't need to kill off. If the avidity for tolerance is higher than the avidity necessary for peripheral activation, then there will be a set of T cells that you have not tolerized that can be activated. So he argued that you want the same avidity for tolerance as for activation. Now, Charlie Janeway had a paper. By the way, before Charlie got into PRRs, he did a lot of experiments. He was a pretty sloppy experimenter. But he did have a paper showing that
Can you say what PRR means? Sorry.
pattern recognition receptor. Sorry, what is PRR? Pattern recognition receptor. Invented by Charlie Janeway. Brilliant. He had a paper showing that tolerance induction in the thymus might require a threshold about 1,000-fold lower than activation in the periphery. I don't know that that was ever repeated by somebody else. Maybe it was, and if somebody out there listening to the video cast knows about that, let me know. But theoretically, you would argue that it should be the same. All right? So, if you're tolerant in the thymus of a particular concentration of something, we're going to come back to that during autoimmunity, right? And you have a cell in the periphery that now expresses a lot more, you will have some lower avidity T cells that have escaped tolerance in the thymus that can now activate in the periphery. However, if that periphery is healthy, what's going to happen to them? They're going to get signal 1 without signal 2 and die. Now, signal 1 without signal 2 is a real simplification, right? We talked about Sinclair's idea and long before the discovery of all these co-inhibitory molecules, that there are co-inhibitory and co-stimulatory molecules, and they're not only expressed on APCs. Right? The co-inhibitory molecules are also expressed on heart and so on. So signal 1, I think Mel was wrong on that one. Signal 1 is a heads up that allows a cell to become sensitive to a whole bunch of co-stimulatory molecules and a whole bunch of co-inhibitory molecules expressed on cells. By the way, it was Sinclair's idea about this that finally allowed me to accept some data that I'd been having trouble with all my immunological career almost. And that is the fact that if signal 1 causes cells to die if they don't get co-stimulation, then how come you can stimulate cells in culture with anti-CD3? Is anybody worried about this besides me? So Sinclair's idea actually solved that for me. Because when you stimulate cells in culture, actually, let me tell you one thing Germain suggested. He said, signal 1 causes a cell to die, but you can overcome that with more signal 1. So if you give a lot of anti-CD3, you can make the cell independent of co-stimulation. Right? That doesn't work for me because it means that if you have a cell in the periphery that expresses a lot of an antigen that is not in the thymus, you ought to be able to activate it with only signal 1. Right? So that explanation doesn't work. But here's what did work, and that is when you put cells in culture, they're on plastic. And you stimulate them with anti-CD3, which is on plastic. Right? Either Cefidex beads or on the plastic. Right? There are no co-inhibitory molecules there. All those co-inhibitory molecules that are there on normal tissues are not there in that dish. So you have your heads-up signal, right? And you turn it on, because you don't have the co-inhibitory molecules on the dish. But you do have co-inhibitory molecules on tissues. So it's that balance.
And you can address that experimentally by putting co-inhibitory molecules.
You could absolutely test that experimentally.
You actually don't get activation.
Has somebody done that? Hang on, let me repeat that so that the video people can hear. So you ought to be able to test that by putting co-inhibitory molecules on the dish. And that's been done?
I would have to look for papers.
You'd have to look for papers. Well, look for them.
Yes.
Okay, I keep asking people to send me those papers. And
Yeah, okay.
actually, Howard Young sent me one. I forgot to send it to you. I will send that to you.
The picture you showed there is of a solid tumor. Is the situation where the malignancy is in an antigen-presenting cell or a B cell any different?
So the question is, this is a solid tumor. Is the situation any different if the tumor were a B cell or an antigen-presenting cell? And the answer is yes and no. So there are, as far as I know, no tumors of functional dendritic cells. There are tumors in dogs called histiocytomas. And I think that they are Langerhans cell tumors. And there's two types. There's one in Bernese mountain dogs, which kills them. And there's one that's prevalent in boxers, I think it's boxers, which has a completely different prognosis. What happens in the boxer, and I know of one Sheltie litter that had it as well, you get a cutaneous tumor, a histiocytoma. It grows, and then it necroses and disappears. Then you get another one that grows, then it necroses and disappears. And then you get another one that grows, then it necroses and disappears. And finally, you don't get them anymore. And there was a guy, Baberham, in Cambridge, England, who was a vet doing, I think, a PhD or a postdoc there. I don't know that he ever published, but I heard him give a talk where he analyzed the difference in function of those two kinds of tumors. And the Bernese mountain dog tumor is a very, very early dendritic cell tumor and does not act as an APC. And the other one, the boxer one, can act as an APC. So here's what I think is happening, right? You've got this tumor growing in skin. It's an unactivated dendritic cell. The dog gets a flea bite or is playing with its litter mates or bumps that skin into something. The dendritic cells get activated. Now they present their own tumor antigens. They get an immune response and they go away. And in the Bernese mountain dog, even if they get activated, I don't even know if they can get activated. They don't act as APCs, so they grow out. Now, a B-cell tumor is an interesting one. I don't know if I've said it yet, but I'm going to say it again, even if I have said it, and that is that I coined the term professional APC, which means I get to define it. Right? Okay. A professional APC is an APC that can turn on a naive T cell. A semi-professional is an APC that can turn on memory T cells. B cells are semi-professionals. If they present to a truly naive T cell, they induce tolerance. We first showed that. Other people have repeated that. It's hard to do because you really need a population of truly naive T cells. If they present to a memory cell, a resting memory cell, they induce activation. There are theoretical reasons why that should be. We don't have time to go into them here, but those are the data. So a B cell is a semi-professional. A dendritic cell is a professional. It can turn on naive T cells. A macrophage, I have no theoretical reason for thinking that a macrophage should not be a professional, unlike a B cell. So if it's a B cell tumor and the cells that can see it are naive, they will be tolerized. If it's a B cell tumor and the cells that can see it have been turned on by some cross-reactive environmental antigen, or maybe the virus that is causing the tumor, right? EBV, for example, then the B cell, if it's an APC, right, if that tumor can actually upregulate co-stimulation, should be able to turn on the T cell. So one prediction might be that B cell tumors are not good at co-stimulating. Now that isn't entirely true because EBV cell lines can be made that are great at least co-stimulating T cell clones, which are memory cells. I don't think it matters whether the tumor is a solid tumor or a liquid tumor. Right? If it's not co-stimulating, it's not going to turn on an immune response. Again, the mere presence of antigen is not enough to turn on an immune response. So this tumor is growing, and while it's growing, if T cells are allowed through there, it's inducing tolerance. Now, eventually, if it's a solid tumor and it gets big enough, and if it's poorly vascularized, it's going to start starving in the middle. Now, starvation death is also programmed cell death, so even that isn't going to turn on an immune response. Until you get so much death in the middle of this tumor that you overload the local scavenging capacity. When that happens, the cells will still die by programmed cell death, but some of them won't get scavenged. And when they don't get scavenged, they go into secondary necrosis, and they'll release alarm signals, and you might get an immune response, right? If there are professional APCs in this tumor that can pick up those antigens, and if there are still some T cells that can see it, right? So by the time a tumor gets to that stage, there might be 3 T cells left that can see that tumor, right? What's going to happen, assuming that you've got professional APCs in there, is you've got enough death to cause alarm signals, right? You overload the scavenging capacity, you cause alarm signals, you activate a local APC. Does anybody have one of these? I'm going to run out, it looks like. All right, I'll try to use it judiciously. That APC is going to go to a local lymph node where it presents antigen, tumor antigen. What have I got? Does anybody have a Sharpie? Throw it down here. Great, thank you very much. Ah, wonderful. All right, so here you have an activated APC presenting tumor antigen. And 3 T cells that can see it, right? So let's say one of them comes in, it gets signal 1, it gets co-stimulated. It makes a small army of T cells with that specificity. They're going to go out into the periphery. And if this has happened, the endothelium in this tumor, because of the death that's going on, is going to be activated. And those T cells, some of them will find the tumor. Now, how many cells can a killer cell kill? Does anybody know? Estimates are 3 to 30 targets before it needs to be reloaded. Right? So you load the gun, you shoot your bullets, and then you got to get reloaded, reactivated. It's not an easy question to answer, but those are the estimates. So let's say your 3 remaining T cells have each made 10, and they go out and kill tumor cells, and each one of those kills 3 to 30. So you've got 10 times 30. So you've killed 300 tumor cells. And now you need to reactivate those T cells. That takes some time. They have to go back into the draining node. They have to find that APC. What's the tumor doing in the meantime? Growing, making millions more cells. Right? You've lost the race. So Steve Rosenberg, America's Mr. Cancer, had a brilliant idea. And that was, if you have a tumor that has tumor-infiltrating lymphocytes in it, right, TILs, that have presumably been activated by this process, but there aren't enough of them, right, pull them out of the tumor, grow them up in bathtubs full of IL-2, and stick them back in. And he did that. And when I first heard him talk, he had a set of slides which I will never forget. He had a patient with a melanoma metastasis in the lung that was the size of a softball. For those of you who are from foreign countries, that's a baseball that is used by women who play baseball. It's called a softball because it isn't quite as hard as a baseball. It's bigger, easier to hit. People think women aren't athletes, right? So they give them big balls to hit. Anyway, softball. He gave this person TILs, and the tumor went down to the size of a baseball. He gave them TILs again, and the tumor went down to the size of a golf ball. And then he stopped. And the tumor grew out and killed the patient. Now, what was wrong with that therapy? Somebody. He stopped. Now, what you think influences what you do. And if you're stuck in the old self-non-self model, then you think that the mere presence of antigen is enough to maintain an immune response, right? Once you can get that immune response going, the presence of the antigen, non-self, should maintain it. And so you think that by getting that immune response going, you can clear the tumor. But it's not true. The mere presence of antigen isn't sufficient to maintain an immune response. You need co-stimulation. You need activated APCs. Now, for the secondary, to keep it going, you don't need professional APCs. You can do it with semi-professional APCs, but you need co-stimulatory APCs. Now, the use of TILs is not the only place in tumor therapy where what you think influences what you do. It's also true for many people who work with tumor vaccines. They're in what I call vaccine mode.
I see that with the vaccine, but what he did, I'm surprised that he followed that logic because it wasn't really about antigen, it was about numbers. I mean, he was growing up the TILs in IL-2 and he was playing a numbers game, which is I can make more of these and kill more tumor cells. It wasn't really about the antigen at that point.
Well, it was. All right, so the question is, he's surprised that Rosenberg followed that logic because what he was playing was a numbers game, right? He was expanding the T cells that could see the antigen. Absolutely right. And if it was a really small tumor, if he'd been able to put in enough T cells to get rid of that last tumor cell, great. But if even the second set of T cells is not enough, to get rid of the last tumor cell, then you need to boost them, because what's going to happen is they're activated, they're going to kill some tumor cells, and they're going to go into a resting memory stage. So you've got the numbers, right? Now, it's also true that they have found, those people have learned an amazing amount of stuff about how to use the immune system to kill tumors. And one of the things they've discovered is that it's not a good idea to use old T cells, right? T cells that you've put in culture for a very long time and have divided a lot. You want young T cells that have longer telomeres and so on. However, my bet is that if they could boost those T cells, once they've gone into a resting memory stage, now they could actually clear that base, that whatever that thing was, golf ball. Okay? The numbers are there. They can pull them out of somebody's arm and see them with tetramers. They can pull them out of somebody's arm and put them in culture and re-stimulate them and show that they either make gamma interferon or kill. But the fact that you can re-stimulate them in culture to make them kill doesn't mean they're killing in vivo. You need to keep boosting them. Okay? So vaccine mode doesn't work here. So what do I mean by vaccine mode, right? Blinkers on. Vaccine, 1 shot, 2 shots. This is a typical thing you do with a vaccine. You prime, you boost. That works for an infection because, you prime, you expand the number of T cells, you boost, you expand them again. That's probably about as good as you're going to get. There's no sense boosting more. Right? They go into a resting memory stage, and when the pathogen comes back, it does the damage that activates the APCs that now reactivates those memory cells. But killing a tumor, killers killing a tumor, unless they're killing a lot of it and overloading the local scavenging capacity, is not going to maintain the response. Because killers kill by inducing apoptosis in their targets. So if you have a small number and you kill, they get apoptosed, they get scavenged, no alarm signals. Bill Heath is in Australia, he's shown that killers killing cells do not induce an immune response to other antigens on those cells. Right? Right? So the killing of a target by a killer does not induce an immune response.
So you would think that if you did a subtotal surgical resection, not so much for the point of resection, but to create damage, then that should allow the co-stimulation to occur, and you should be able to completely get rid of the tumor that way.
So the suggestion is you would predict that a surgical resection, not for the purpose of clearing the tumor, but for the purpose of causing damage, would actually cause the tumor to go away, or at least to get an immune response. Sure. But in order to do that, you have to not clear the tumor. Right? So most oncosurgeons actually try to get all of it. They're going to cause some damage, absolutely. They're going to activate local APCs, absolutely. But there's no tumor antigen left.
But there's a lot of times where they can't get rid of the whole tumor, right?
So there are times when they can't get rid of the whole tumor. And what happens then? Prediction. Right? So first, I was told just 2 weeks ago, I can't remember where I was talking about tumors, by a surgeon, that if you go back in the literature, you find that dirty surgery, meaning where they don't get the whole tumor, gives you sometimes a better prognosis than clean surgery. So I would say that's because the tumor antigens are there and actually getting presented by the APCs. But by itself, that's only a prime. Right? If you want to get rid of the last tumor cell, you have to keep boosting. When you cut that tumor and you leave tumor antigens, if there are still T cells that can see it, you will get a response. It'll come up, peak at day 7, and be gone by day 14 or day 30. Right? If the dendritic cells, remember the life of a dendritic cell estimated are 3 to 21 days. So you activate those dendritic cells. They're now expressing tumor antigens. Not expressing, but presenting tumor antigens. They're dead in 21 days. A few days after that, your last T cell that needs to be restimulated won't be restimulated. It's going to go into a memory stage. You need to keep boosting. So there was a paper that I sent out for you all to read, showing that if you do damage in a tumor, you can, actually, we don't know if it was an immune response, but if you do repeated damage in a tumor, you can often clear them. Who did that? Coley. Back in the 1890s, when we didn't know squat about the immune system, Bill Coley was injecting a nasty mix of bacterial toxins into tumors. Now, he had a very nice paper in 1898, which I fell across long before we could find papers online, and I was in the bowels of the NIH library. Following references back, I came to this paper in 1898, where he was injecting toxins. So the story of Coley is kind of interesting. Coley was a surgeon in New York, and he ran in the right circles. He was friends with the Rockefellers, and one of the Rockefeller kids had a fiancée who got a tumor. And Coley's a surgeon. He says, no problem, I'll just cut it out. So he cut it out, and it recurred, and she died anyway. So he got really interested in tumors because of this. Right? This was a surgical failure. And he started looking at how to cure tumors. And he had a patient who had a sarcoma of the neck, on the neck that got an infection with a bacterium called erysipelas. And erysipelas is a pretty nasty bug. And the tumor went away. It was a pretty large tumor, and it went away. So he started injecting people's tumors with erysipelas. And unfortunately, he killed as many people as he cured. So he decided that was probably not a good thing to do. So what he started doing instead was making a mix of the toxin from erysipelas and the toxin from another bug whose name I can't remember, but it's in that paper. Sorry? A bacillus of some kind. Okay. And that mixture was able to make tumors go away. He didn't cure everybody. He did about 30% of his patients that had sarcomas. And one can guess that the ones he didn't cure were the ones where he could get rid of the tumor he could inject into, but then there would be metastases in other places that he couldn't get to. Now, what was important about what he was doing? A, he was causing damage in the tumor. B, he was doing it repeatedly. Okay. He was those two things. He was locally causing damage, and he was doing it repeatedly. Now, what's important about local causing damage? A, you turn on the immune response, right? The dendritic cells there actually pick up tumor antigens and present them. But also, by causing damage in the tumor, you activate the local endothelium in that tumor so that when the immune system gets turned on, the lymphocytes can actually get to it.
Doesn't he also point out in the paper that he did
I'm sorry, I can't hear you.
Doesn't he also point out in the paper that he did injections remotely? Remotely from the tumor?
Yeah, that there was a
He
system.
did. He did. All right, so didn't he point out in the paper that he also did injections remotely? And he did. And there are people today still doing that. There's a company in Canada, Don McAdam, I think is the head of that makes a Coley's-like toxin. They call it Coley's fluid because toxin isn't a nice word. And they inject remotely. They inject to the point of getting fever. So for them, it's very important to get fever. Now, fever is something, at least in experimental cultures, that will activate dendritic cells. So by causing systemic fever, you're not only going to activate systemic dendritic cells, you're also going to activate the ones that are in the tumor. It's, in my opinion, a lot better to do it in the tumor.
I was kind of wondering, was this like the first chemotherapy?
I guess you could call it chemotherapy.
And it was before X-rays? Because he doesn't mention an X-ray at all in the paper.
Does anybody know when X-rays were invented? 1930s? 1905?
The Nobel Prize is given.
So before X-rays.
So I mean, I agree with what, I can't see him.
We
But
can't hear you.
yeah, he showed a systemic, or he made an argument for systemic effect, that you could inject remotely and have action
Right.
even on the tumor.
If he got fever. Now, there's a paper in dogs where they use BCG, which is also a bacillus. It's actually a variant of bovine tuberculosis, which is still used in bladder cancers. They infuse it into the bladder and they roll the person so that it gets all over the bladder. There's a venereal tumor in dogs that the incidence is going down because dogs don't mate wildly as much as they used to in the past because they're on leash and they're neutered. But it's a venereal tumor where the tumor is transmitted sexually. It's not a virus that's transmitted, it's actually the tumor. That tumor always has the same MHC type no matter what dog you find it in. Right? So even though it's allergenic, even though it expresses MHC, it's transmitted sexually and it grows. And there was a paper using this tumor where they got dogs that had the tumor, and they either injected BCG into the tumor or they injected BCG on the other side of the dog. And they found that it was most successful if they injected it into the tumor. So I think there are two reasons for that. One, you get activated APCs that actually present tumor antigens. And two, you activate the local endothelial cells so that once you've turned the immune system on, the lymphocytes can get there. Okay, back to, go ahead.
I think, Coley's paper goes into 2 points. I completely agree with the...
If it's quick, you can say it from your seat. If it's more than 3 sentences, you have to go to the microphone. It's good exercise.
Hi, Paul. I'm saying that I think Coley's paper talks into 2 points that he agrees with the just creating the danger. You don't actually need the surgery because now there's evidence that just radiation activates the immune system. There are lots of people out there with that.
So how do you
and
think,
I think
wait, before you go on, how do you think radiation does that?
because you can irradiate the mass and measure the activation markers.
No.
And all
How do you think the radiation, by
It's
what?
breaking because the radiation is just creating the danger by killing cells. That will automatically activate the system by just killing the cells.
But most radiation causes apoptotic death.
Yeah, but lots of them also do cause
Some of them.
some. Yeah.
I think radiation will work when you kill so many cells that you overload the scavenging capacity.
Yes, I mean that's why
Except
it doesn't
maybe
work.
proton
Yeah.
therapy. I think proton therapy looks like that's necrotic.
Yeah.
You
Yeah.
stick protons in cells.
So,
All right, go on.
So I think it agrees on that in the sense that you just need to create that danger, and I think the fact that you can inject the toxin remotely and get a response because most of those toxins do have receptors, so they can go everywhere and then bind the cells and enter and kill the cells, and that's how some of the ideas behind the oncolytic virus for tumors work, because most tumor or most viruses, just like poliovirus, that's treating the glioblastoma, because they can use that. You inject it remotely, and it finds the, because most cells in the brain do express the poliovirus receptor, and so you can manufacture that, and just giving that virus, you cure the glioblastoma. So it also works in the sense that you do have most toxins or most viruses will have a receptor that even if you give it remotely, it may find a target away from the injection site.
Except that, at least in the Coley's case, and also the Canadian more modern version of that, if you don't get fever, you don't get a very good response. So even though the toxin may be going to lots of tissues, and actually that's not a toxin you want to use, right? Because by the time you get enough on the tumor to kill the tumor, you've also killed everything else. For them, it's fever if they use it remotely.
That means you're having inflammation.
You're having inflammation. But it doesn't necessarily mean that the toxin is binding to the tumor cell, necessarily.
Yeah, not necessarily, not only tumor cells. Also, that's what people can die because it may bind also normal cells.
Yeah, it also binds normal cells. So that's not something you really want to do, unless you can get it into the tumor. If you can get it into the tumor, then using a non-specific toxin is fine. So, but you said something else that's really important, that radiation, so people know now from mouse studies that one of the ways in which radiation works on tumors is that it activates the immune system. So if you don't have an adaptive immune system, radiation isn't nearly so good. Right? You need to activate the immune system to have radiation be really effective. So that brings us to another paper we read, which is what aspect of the immune system do you need to activate in order to get an effective anti-tumor immune response? So most people work on trying to activate CD8 T cells. But we had a paper to read that said that they don't work alone. Anybody read that paper? Anybody want to tell me about it? Nope.
Is that the dynamic activation?
I'm sorry?
Was that the dynamic activation from your?
I can't hear you.
Sorry, was that the dynamic activation one from your?
It was one where they showed they need CD4 help.
Yeah, just the
Don't do it from your seat.
1998 Rich.
No.
I'll say.
Who wants to tell me about the Restifo paper showing that you need CD4 help? Okay.
I guess what they did there is I have some questions about that paper, but
Oh, I had lots of questions about that paper too. That's okay.
they put beta galactosidase in tumors, and
So why did they put beta galactosidase in the tumor?
because it elicits normally a pretty good immune response.
No.
No? Okay.
Nope. By itself? Nope. So the reason, so they have a tumor, they transfect it with beta-gal. What is beta-gal? Where does it come from? It comes from bacteria. It is a very foreign molecule. So what they were doing here is creating an experimental tumor antigen, an antigen to which the immune system ought to respond strongly. Okay, go on.
Okay, and and then
And it's growing.
they made beta-galactosidase CD4 positive T cells, or beta-galactosidase responding reactive CD4 positive T cells.
How did they do that?
They did it by selecting for clones that express IL-2. And
How did they activate them?
how did they activate them?
How did they activate? So they have a mouse that's got a whole bunch of CD4 T cells. They want to make T cells against beta-gal. The tumor itself isn't going to do that. So how did they do that? Anybody? Inject them with the antigen? No. By itself, that wouldn't give you an immune response. Unless you bought it from Sigma and it had LPS, right? They used a virus. They made a virus expressing beta-gal. So they immunize the mice with this virus expressing beta-gal, and then they get CD4 T cells, and then what?
And then they selected among all the clones for those that expressed IL-2 and interferon gamma. And, okay, so, I mean, my original statement, I thought, I was a little bit confused about this. So the beta-galactosidase in the virus induces a good immune response, but not in the tumor.
Correct.
Right? So it's immunogenic in that sense, or antigen.
It's immunogenic it's the virus, right?
In the
virus is
virus,
doing
but not
damage,
in the
da-da-da.
tumor.
The tumor
Right.
doesn't. Yeah.
And so they, so then they did some studies where they claimed, I guess, that they deleted all the CD8 positive T cells.
Hang on. Start with. Do they need these? Do the CD8s get rid of this tumor without those CD4s?
No.
No. Okay. So that was the first important point of that paper. That was, in fact, the most important point of that paper. That if they don't put in these tumor-specific CD4s, the mouse does not clear the tumor. CD8s by themselves don't do the job. They need help.
Okay, so then
Now they made a second point.
Okay.
Go ahead.
So then at some point they said they deleted all the CD8 positive T cells and showed that they couldn't clear the tumor that way, but they didn't really say how they deleted all the CD8 positive T cells in the paper.
Yeah, okay, so they made a second point in the paper that I actually think they didn't really show, and that is that the CD4s don't do it by themselves. And the way they did that was to put the CD4 T cells into mice that are class 1 positive, negative, MHC class 1 negative. Mice like that don't have CD8s. So they're putting these activated anti-tumor CD4s into a mouse that doesn't have CD8s. And they don't clear the tumor. And from that experiment, they conclude that the CD4s by themselves can't do it.
Okay.
Now, I don't agree with that. The thing about class 1 negative mice is they can reject class 1 positive cells using NK cells and other ways. So when you put CD4 T cells into a class 1 negative mouse and you don't get an effect, you don't know if it's because you need CD8s or because your CD4 T cells got rejected, and they didn't look for that. So that was a problem with that paper. But the important point about that paper that wasn't problematic is that in the absence of CD4s, the CD8s didn't work. So people who are making vaccines for tumors using specific peptides that can be seen by CD8s need to read this paper. Right? That CD8s by themselves, without help, are not going to do it. So how does a CD4 T cell help a CD8? That's the Ridge paper. Sorry? IL-2? Nope. Turns out nope. Even though Rosenberg uses IL-2 as a way of trying to help the CD8s, I think what's actually happening in his experiments where he gives TILs and IL-2 is he's giving enough IL-2 to get vascular leakage. And that will allow cells to go into the tumor.
So it causes disease to upregulate the B7 costimulatory.
It causes, okay, so CD4s help CD8s by what we called conditioning the APC. And Jonathan Howard later invented the term licensing, which Antonio Lancevecchia used when he wrote a News and Views about this paper. So what happens? There are two ways. We started this paper, there were two ways. So you have an APC. What was already known is that the CD4 and the CD8 need to see antigen on the same APC. They don't have to see the same antigen, but they need to see the antigen on the same APC. So there were two possibilities for that requirement. One, if this is the CD4 T cell, and this is the CD8 T cell, one possibility is that the CD4 makes IL-2, and that goes over to the CD8. Another possibility, which was first suggested by Kevin Lafferty, remember Kevin Lafferty? He's the one who invented co-stimulation, Lafferty and Cunningham, is that the CD4 does something to the APC, which now allows it to turn on the CD8. Long story short, this is the process, not this one. In the Ridge paper, that was one of 3 papers actually from 3 different labs showing that CD8s anti-tumor, or CD8s, in this case anti-transplant, needed help. One of the things that's important about this paper and that is not usually picked up is that these were memory CD8s. These are not naive CD8s. And what it says is that even the memory cell needs help. So when you put in TILs and you purify CD8s, you need to give them some help. And when you boost them, you need to give them some help. Okay? CD8s by themselves aren't going to do the job.
Just to understand the difference between the difference between Lafferty's model and Ridge is what Lafferty would say they have to be on the same APC when the CD4 activates the APC's ability to activate CD8, whereas Ridge would say they don't. Or
No, so I don't think there actually is a difference between Lafferty's model and Ridge. So both of them, I mean, Lafferty's was theoretical, Ridge was the data. By the way, John Ridge was a technician. This is a first author, I think, Science paper. I can't remember if it was Science or Nature now. It took him 4 years to do this. They don't have to be there at the same time. That's what's really important about this. The CD4 licenses the APC. You can now get rid of the CD4s, and those APCs will be able to turn on the CD8. And what's important about that disjunction in time is that, you know, the CD4 against one antigen and the CD8 against one antigen are both rare cells. The chances that they're going to meet the APC presenting that antigen as they're going through the lymph nodes, right, at the same time is really low. Even with this disconnect of time, it's amazing that they meet the same APC. Now, Ron Germain's group actually had more data on this, but I already gave you 8 papers, so I figured there were, you know, I could have given you 50. Okay? They showed that when the CD4 turns on this APC, the APC somehow actually calls to it the CD8s. It starts making some kind of chemokine that draws CD8s to it. So when the CD8s are going through a lymph node, those APCs are the APCs that they're most often going to look at. Okay? So if you want to make a good tumor vaccine, you need a vaccine that turns on CD4 cells and that turns on CD8 cells. Now, there is also evidence that CD4s can do it by themselves.
licensing stable?
How long is the licensing stable? I have no idea. I would guess for the life of the dendritic cell. But we haven't done that experiment. And I don't know that, I'm trying to think of how you would do that experiment. I mean, in vitro, a lot of this was in vitro. Some of it was in vivo as well, but in vitro you can't really say anything about how long because the culture conditions are never going to be the same as in vivo. I suppose you could have, you could do this. You could take male dendritic cells, which are expressing HY, and either allow them to interact with a CD4 T cell or not, and then inject them into a mouse that has no CD4 T cells. Say rag knockout. And then at various times you could add naive CD8s and ask, do they get activated? You could do that. It's not an easy experiment, but you could do it. I don't think anybody's done it. Okay, so if you're making an anti-tumor vaccine, you want to at least turn on CD4s. Is there any evidence that CD4s by themselves can do it? Yes. So Ainhoa Perez Diaz, another paper I could have given you to read. I know is a Basque researcher here at the NIH. Not only is she a great anti-tumor researcher, she's also, I don't know, black belt 9th Dan or something in Taekwondo. She represented the U.S. In some tournaments in Korea. Kind of amazing woman. Anyway, she showed that CD4 T cells, these were TCR transgenic T cells called Marilyn. She also used CD8 T cells called Matahari. Matahari was a World War I spy, so an anti-male killer. And Marilyn is from Marilyn Monroe. She's an anti-male helper. And she showed for 6 different tumors from different tissues that were carrying HY, right, so male tumors, that the CD4 T cells alone could cause tumor rejection. Took her 4 years to get that paper published. You know, she went down the list. Nature Medicine, da-da-da-da-da. It was finally published, I think, in Circulation Research. Nobody wanted to hear this. And then a few years later, Jim Allison's lab and Nick Rustifa's lab had 2 papers back-to-back in the Journal of Experimental Medicine saying exactly the same thing. So that's 3 different labs showing that you can clear a tumor in the absence of CD8 cells if you have good CD4s. So right now, I think the tumor eradication field is really focused on CD8s, but I think they need to start focusing on CD4s as well, and a few people are. A few people are focusing on CD4s. Okay, so if you do damage to a tumor sufficiently often enough, you can sometimes get rid of it. In fact, you can usually get rid of it. It's not necessarily the metastases, although sometimes you can get rid of the metastases as well because you turn on an immune response to the primary tumor, and now that immune response can also clear metastases. However, if the metastasis is happy, meaning its endothelium is not activated, then even the activated T cells might go right on by, right? Because you know you need it activated. I keep pushing this. You need activated endothelium so the T cells will slow down and roll and stop and go out into the local tissue. So you need to do something to the tumor. So vaccinating somebody in the arm when they've got a tumor somewhere else may not work very well unless you also do something to the tumor. So you need damage to start the immune response, you need damage to maintain the immune response, and you need damage to bring the immune response to the tumor. Now, if the tumor is dying a lot, it's a big solid tumor and it's overloading the scavenging capacity, then you've got enough damage there and you don't have to do it. But if it's tiny little metastases in the lung, you know, you might want to give somebody an inhaler with something nasty. There's a very good example of that in another paper I thought about giving you to read, which is not about tumors, but which shows that you need to have damage in the tissue to get an immune response to get rid of the tissue. And that is a paper by the group in Heidelberg, where they have, again, a TCR transgenic T cell. So its T cell receptor is against H2KB, which in the mouse is a major histocompatibility class 1 antigen. So a really good target for CD8 killers. And they also have another mouse which expresses, in fact they have several, expresses KB. Okay. And they have several mice that express KB. They have one that expresses it only in the tip of the tongue. They have one that expresses it in the skin. They have one that expresses it everywhere. They have one that expresses it in the liver. So what they did with the one in the liver is kind of interesting. They activate these guys. All right, so they now have a population of activated CD8 T cells. And they infuse them into the mouse that expresses KB. Right? A lot of them. These are hot shit activated CD8s. If they test them in culture, they kill. Can anybody predict what happens to the liver? It's the target. Who says it gets rejected? Who says it doesn't? Sorry? Does the liver? So the liver is carrying the target for these activated CD8s. Does it get rejected? Nope. Nope. Perfectly fine. It is totally perfectly fine. Unless they give that mouse a virus which is liver tropic, or they give it a really small dose of carbon tetrachloride. Not enough. Carbon tetrachloride hurts the liver. It can destroy the liver. But they give a really, really sublethal dose of carbon tet and these CD8s, now it wipes out the liver. Right? So simply activating the cells is not enough. You need to do something to the tissue, or if it's a tumor, you need to do something to the tumor. Okay. So, there are people who have understood that you need to keep this immune response going. All right, this is not just theoretical anymore. We have 2 papers that we read, aside from Coley, where the authors thought it was important to just keep boosting that immune response. One of them was the paper by Ben Dandy. Yeah, anyone want to tell me about Ben Dandy? You've all been burnt. Nobody wants to get up to the microphone anymore. You've done one.
I've done one.
You've done one. Come on. Okay. Quick.
Okay.
We have a half an hour left.
All right, so he basically had 33 patients, and they'd all experienced their first relapse of follicular lymphoma. Mm-hmm. And he used rescue hybridoma to isolate the patient and tumor-specific antigens from tumors.
Which is? What kind of antigen? So does everybody know what kind of antigen a lymphoma has a lot of? It's a B cell that makes antibody. Right? So what Ben Dandy did was to isolate the antibody that each individual tumor was making. So this is patient-specific therapy.
So he isolated those proteins and then he coupled them to the keyhole limpet hemocyanin.
Yep.
And injected it sub-Q with GM-CSF as adjuvant. And he did this 4 injections once a month, and then 1 boost 2 months later, and then 5 more boosts every 3 months. And 80% of the patients showed a vaccine-induced response. 2/3 of these showed a humoral response that slowly declined with subsequent vaccination.
What happened to the tumors? Let's cut to the chase here. What happened to the tumors?
Well, they were basically, they got no relapses during the vaccination period. So basically they had regression and essentially I guess the question at the end of the paper is, did the patients die when they stopped the trial?
Well, no. Some of them. So, if you're doing a personalized vaccine, it's hard to know what to use as a control, right? Every vaccine is different. Every single person's vaccine was their own idiotype, the own antibody that tumor was making. So what they invented was this idea of using each patient as their own control. So this is actually the use of a vaccine which has been around for a while. This vaccine was invented by a guy named Ron Levy at Stanford. And they take the tumor, the lymphoma, they hybridize it, they get hybrids that make the antibody. They stick it to KLH and an adjuvant, and they inject it. And their first protocol was 5 injections. And their phase 1 trial was pretty astonishing. They had, I think it was 9 patients. Now you've got to realize that phase 1 is a safety trial, right? You've got people who are going to die. They've given their bodies to science, and you're really asking the question, are you going to kill them faster than they would die anyway? And out of the 9, I think 7 went into remission. That's amazing for phase 1. However, 5 injections, right? And some of those people then later relapsed. So Ron Levy had a postdoc named Larry Quack, another famous oncologist, right? And Larry Quack had a postdoc named Maurizio Bandandi. And Maurizio Bandandi decided to try using the same vaccine and just continuing to boost. So they treated 25 people. 20 of them responded, 5 didn't. Now, from earlier papers from that lab and the great-grandfather, Levy's lab, what they've learned is that if somebody still has minimal residual disease, they probably won't respond. And that's because these are not professional APCs. These are B cells expressing antigen. And if you still have minimal residual disease, you've got a lot of B cells expressing this antigen. And in my opinion, they're going to induce tolerance. So you need to get a person who has been treated and no longer has residual disease. So they treated 25 people, 20 responded. Of the 20, 18 had not relapsed by twice the amount of time that they relapsed before. So what we know in lymphomas is if you treat somebody and they relapse, they're going to relapse again quicker. And the reason they relapse is they have a few cells that are kind of resistant to the chemotherapy. And of course now those are the ones that are growing out. And so any mutations there that makes them even more resistant will grow out. And so what happens to lymphoma patients who relapse is they relapse, and then they relapse again, and then they relapse again, and then they relapse again, and pretty soon you can't treat them anymore. So what they found was that 18 out of the 20 people who responded had not relapsed by twice the amount of time, at least, that they had relapsed the first time, and by twice the average relapse time. Nobody relapsed while they were still giving the vaccine, which was 2 years. One guy died at 2.5. He relapsed. And that is as far as I know about how far that paper went. I was going to try and get a hold of them and see how those people are doing now, but I didn't. Maybe I'll do that for next week. See how those people are still doing. So 80% response. And if you look at the paper, most of those people are out 5 years. That's pretty good.
But they're not being vaccinated anymore.
No. But one guy died at 2.5. So what it says is if you can keep vaccinating long enough, you can clear the tumor. Now, this tumor does not have so much of an access problem because it's a lymphoma, right? It's in lymph nodes and places like that where the immune system gets to anyway. Okay? If this were a solid tumor and you were vaccinating and you needed to boost and boost and boost and boost, you would also need to be doing something to the tumor, right? The easiest thing is to boost in the tumor, but... Okay, so that was one way of keeping the immune response going, which is just keep boosting. Another way that people tried was to link the T cell receptor to an internal piece of a receptor for co-stimulation. So this was the Carl June Carr paper, right? So what do? They make T cells. They get T cells from the person who has a tumor. In this case, it's a leukemia, which is also a B cell tumor. But unlike a lymphoma, leukemias are what they're called wet tumors. Lymphomas are quite solid. It's again a B cell making antibody, but they didn't target, in this case, the antibody. What they targeted was a molecule called CD19, which is on the surface of just about all B cells. Right? So what they're going to do with people that have a leukemia is simply wipe out their B cells. Right? Their normal B cells and their leukemic B cells. Unlike the Bendandi vaccine, which is targeted just to the tumor B cell. So what do? This is the tumor. They make a T cell that has an antibody against CD19. So one of the ways that tumors can escape the immune system is they lose MHC. And we know that killer cells need to see peptides in MHC. So any tumor that mutates that loses its MHC can no longer be killed by a CTL. It can be killed by NK cells, but not a CTL. So what they did is they made an antibody against CD19. So this is specific for CD19. And they attach it to a CD8 alpha hinge, and then they attach it to a transmembrane region of CD28. In fact, let's do it this way. Here it is. CD8 alpha. Right. Transmembrane. CD28, does everybody know what CD28 is? It's a receptor for co-stimulation on T cells. Okay. And then internally, they attach it to an internal activating region of CD28. I can't remember which direction they did this now. And to CD137, which is also known as 41BB, which is a receptor for another co-stimulatory molecule. Right? So 2 receptors for co-stimulation, the CD28 transmembrane, the CD8 alpha, and CD19. Now what that means is when this thing binds and the antibodies patch because of the binding, it's going to bring these together, and it's going to activate not only this co-stimulatory receptor, but also this co-stimulatory receptor. So what they've done here is they've removed the need for co-stimulation on the target cell. The simple binding, or at least theoretically, the simple binding of that receptor to the tumor should be sufficient to keep reactivating that T cell. So that was their way of keeping the immune response going. Right? They know that you have to keep it going. So what happened? They have people that have this tumor. They infuse them with the T cells. The paper had 14 patients. What happened to them?
8 of them had some response.
I'm sorry?
8 of them had some response.
Actually, I think for the Carr paper it was 4.
4
Four
had a complete response.
4 had a complete response, right. 8 of those 14 responded, 4 of them had a complete response, meaning they got rid of their tumor.
About a fourth of them did.
4 of them, 4
Yeah.
out of 14.
1/4 of the patients.
A little bit more than a fourth,
Okay.
4 out of 14. A little bit less than a third, a little bit more than a fourth, whatever that is, 29%. Cleared the tumor. Now they noticed something about the ones that didn't, and that is that the T cells didn't persist. So they had a figure. So each one of these is a patient. Okay, whatever. And they looked at the expansion and persistence of those CAR T cells. And they found that on the whole, can't remember exactly how it went, the complete responders had T cells that persisted. There was also one partial responder that had T cells that persisted. But everybody else, whether it was a partial responder or a non-responder, the T cells went up maybe a little or maybe a lot, but they disappeared. So for some reason, the CD28 or the CD137 was not working in some people. Right? It worked in 5 out of 14. And in 4 of those 5, it helped to clear the tumor. In the other one, they got a partial response. Now, I don't know why this would be, and they don't know either. They looked at all kinds of characteristics from those people to try and figure out why the T cells persisted in some and didn't persist in others. And they couldn't find anything. But in reading up on CD137, I discovered something that may be a hint. And that is that there's a guy named Kwan who showed that if he made CD137 negative mice that were B6, so B6 is black and is H2B, or BALB-C. BALB-C is white and H2D. They're different in a lot of other ways. Very different mice. That the CD137 negative B6s had an upregulation of inflammation. The CD137 negative BALB-Cs had a downregulation of inflammation. So the same molecule in different environments can behave differently. And we know that from other studies. It can, in some cases, it can upregulate Tregs. In some cases, it upregulates T effectors. Right? The ligand is on APCs. The ligand is also on normal tissues. And depending on what tissue it's on, it can cause a different response. And depending on what level it's at, it can cause a different response. And the way it seems that works is that the 401BB and its ligand are both on the membrane, and they cause each other to downregulate. And so if you now have another cell which has the ligand at low levels, you still have a lot of downregulation. If it has it at high levels, now it holds it to the surface. So that's one way in which it could be possible to titrate the signal. I don't know that that's it, but one thing that June and his lab might want to do is see what's different between B6 and BALB-C and see if that is also the difference between the patients that respond to this CAR and, or where they can persist and the ones where it can't persist.
I had a question.
Make it quick. We have 7 minutes left.
All right. Didn't they use a lentivirus to infect the T cells? Is it possible that in some of the patients that their T cells were just not as tolerant of the virus?
I don't know. No, it's the same. I don't know. Possible.
Just
I don't know how much of that virus is left in those T cells or how much of it is expressed. So you can imagine that some viral antigen, it's a lentiviral vector. So I don't know how much virus is left and how much is expressed. If there is something expressed, you could imagine that some people reject those T cells. It's possible. It is absolutely possible. I don't know that.
On the CAR T cell, there's a viral particle that acts as a linker, and there are antibody mounted against those viral particles. But I don't think that's the main problem with CAR T cells. And we know that there are antibodies made against, because when they make the CAR, there's a linker, which is a viral particle. I mean, now they've taken that viral particle out.
That's okay. We don't have the time, but go ahead.
So they've taken that
They've taken the virus out.
virus
They
out.
no longer have that.
Yeah, but the problem that has caused some death in the CAR T cells still remains the systemic cytokine that nobody's able to solve.
All right, so they've taken the virus out.
So that there's no
No more antiviral response. Okay. But the T cells don't work as well either.
They don't work, but they still face this problem, major problem of systemic cytokine.
You know, you really need to go to the microphone when you do this.
No, I'm done.
Okay. They've taken the virus out. The T cells don't work as well, but they still release cytokines. Okay, speaking of cytokines, all right, really quick. We got 5 minutes. I don't know why this one went so long. We have 5 minutes, unless you want to stay. But you can't stay long anyway, because those people have to go home. The other thing people have done, rather than try and maintain T cells by giving T cells or by boosting, is to try and upregulate the natural activity of those few T cells that are there already, trying to get rid of the tumor. Right? And they do that by what they call checkpoint blockade. So remember those co-inhibitory molecules? CTLA-4, et cetera, which one did we read about? Sorry?
PD-1.
PD-1. PD-1 is a molecule which, if you stimulate it, the cell goes into apoptosis, or at least sometimes. So they wanted to block that, to see if the normal apoptotic signals that are probably given by co-inhibitory molecules, could, if you block those, could you now allow the person's own native T cells to expand and get rid of the tumor? And the answer is yes in some cases. Right? So they had 33 patients. I can't remember exactly now how many responded. I think it was, actually, they had 33 patients to start with. They treated 25. I think it's going to be a little bit like Bendandy, 18 or so responded. They had, in this case, 8 complete responses. So 8 of those people cleared their tumors. So everybody's doing about the same, right, with these various treatments. One of the differences is that some of them have worse side effects than others. So people who get checkpoint blockade, there's actually a paper about all the adverse side effects of checkpoint blockade tests. They get, a little under half of them get skin rashes. They get diarrhea. Some of them lose their thyroids. Some of them, the thyroids return function. Some of them don't. It's permanent. A few people get uveitis. When you start doing checkpoint blockades, right, you're blocking the ability of the co-inhibitors to work, which means you now have this possibility of inducing autoimmunity. And autoimmune problems are showing up. In mice, you can kill mice with checkpoint blockades if the mice are autoimmune prone. Okay? So right now, patients that have autoimmune diseases are on the whole not in these trials. So if you have an autoimmune disease and you get a tumor, you're out of luck. Okay? Whereas something like Bendandy, where they're making a vaccine that directly targets the tumor, they don't have that side effect. Okay, so what are the take-home messages from this session? You need to turn on an immune response. You need to maintain the immune response, right, until the last tumor cell is gone and longer. You need to do something to the tumor if it's not a lymphoid tumor of some kind. You need to do something to the tumor to get the immune response to go there. And the one thing we haven't discussed, and we'll discuss later, is you need to make sure that your immune response is of the right immunological class. And we'll talk about that when we do class, which is next week. All right, in the few minutes we have left, are there any other questions? Yeah. Why do MHC1 deficient mice reject MHC positive cells?
Why do they reject CD4s?
Why do they reject CD4s? They're going to reject all kinds of cells that come in. Right? So people used to think they won't reject anything because they don't have a lot of CD8 cells. However, there's evidence from our lab and other people's labs that CD4s can also reject tissues. So they do have CD4 T cells. They also have NK T cells. Cells that are MHC1 positive may not do well in them. That's all I know. Go.
How does CD4 alone kill the tumor?
Ah.
B cell help?
How do CD4 T cells kill a tumor? Well, that is Ainoa's second paper, which he's also been trying to publish for 4 years, and nobody wants to hear it. So quickly, the take-home message is that it changes the phenotype. The CD4s change the phenotype of the tumor-infiltrating macrophages. That's all I'm going to tell you. I mean, somebody accept this paper. Yeah? Nobody wants it. The reviewers don't even, the editors don't even send it for review sometimes because it's been done in immunodeficient mice. Now, Ainoa's argument, and mine too, is if you want to show how a CD4 clears a tumor in the absence of CD8s, you have to do it in the absence of CD8s. You can't do it in normal mice. Because if you do it in normal mice, everybody will simply tell you it's the CD8s. And so there, there's, what do? You do it in the only way you can do it. And then the editors tell you that that's not a physiological system. I'm really sorry. I don't know what to do about that.
But can't you just deplete the CD8?
It's the same. You deplete the CD8s. It's not quote, normal.
You have to prove that you have complete. They'll always say that.
Yeah, and by the way, we tried depleting CD4. What he just said, you have to prove that you've depleted the last CD8. And by the way, it's really hard to do. When we tried depleting CD4 T cells, we took the mice, we thymectomized them. We depleted the CD4 T cells using 2 synergizing antibodies, and we waited. And they come back in the absence of a thymus. Now, they're oligoclonal, and here's what I think is going on there. Right? The death is probably by ADCC, right? NK cell mediated death. And at the time that you deplete them, there will be some T cells that are in germinal centers. And I don't think there are NK cells in germinal centers. And so those T cells are actually protected. It's really tough to completely deplete.
So this goes back to the beginning with the...
Coley?
Yeah.
Oh, great.
How do you know? So one prediction would be that if fevers can induce this, wholesale activation of dendritic cells, and that's really what's responsible for the effect, you would expect people with chronic febrile illnesses to be prone to autoimmunity, to spontaneous abortion, things like that. Are you aware of any clinical data or preclinical studies?
That is really interesting. Or people who have malaria, right?
Anything that gives you a chronic febrile illness, right? Viral or otherwise.
That's an I know of no data. You do this a lot. You have interesting, yeah? This is a guy that does not think in a box, huh? Yes? Every single normal pregnancy is associated with fever. Okay?
So that is telling you that if you have that innovation, it causes some sort of system. Because even if a woman delivers normally, at the time of delivery,
Oh, at the time of delivery, you have fever. Oh, that reminds me of the other thing I forgot to say that I wanted to say at the beginning here. So several people have written to me about RH disease. Right? If the danger model says that you don't reject fetuses because they don't look dangerous, Why do women respond to RH? And this one's interesting. I think, again, God is in the details. Right? The problem with RH disease is in the second pregnancy, not the first. Women get primed at birth. Right? Birth is dangerous. You have ripping, bleeding, surgeons with knives, dangerous time. Right? And the medical profession knows this. They know that they get primed at birth because if they have an Rh negative woman who's carrying an Rh positive child, they give her anti-Rh antibody right around birth to cover up those antigens so that she doesn't respond. Right? It's not the pregnancy that primes. It's birth. So if there's fever at birth, great. You know, you've got a lot of stuff happening. You actually want to turn on your immune system to get rid of infections. You've got a placenta to get rid of that you may not get rid of immediately. It could, you know, yeah, I'm not surprised. Any other questions? Okay, next week, how do we regulate class? Self-antigens than the normal tissue. So these ought to be rejected, and yet they're not. And when Ephraim first suggested that this was a problem with the self-non-self model, I patted him on the head and said, you know, tumors are immunosuppressive. And there are a lot of people that still say that. You know, there's the myelo-derived suppressor cells in tumors. There's the T regulatory cells in tumors, and so on. Tumors are immunosuppressive. But we know that the immune system can get rid of tumors. So why doesn't it? So for a while it was thought that it does get rid of tumors. It gets rid of most of them. You know, the idea when I was a graduate student in the 70s was that we get a tumor every 3 minutes or so, and the immune system gets rid of them. And when we get a tumor, when a tumor actually grows out, it's because the immune system couldn't get rid of it. And that was a theory until about the late 70s when the nude mouse was discovered. Has no T cells, has no thymus. Actually, nude mice have a few T cells from their mothers. Just for those of you who work with nudes or ever plan to work with nudes, be careful. Because nude mice in most colonies are bred from heterozygous nude mothers. And that's because nude mice have no hair, and they're not very good at keeping temperature control, and so they have trouble during pregnancy. And so the way people breed nude mice is they have heterozygous mothers, which have hair and thymuses, and homozygous fathers. And so half the litter is nude and half the litter is hairy. Well, depending on what colony and how they're bred, nude mice have T cells from their mothers. So be careful when you find nude mice. So for a while people thought nude mice had T cells because they were growing up in the omentum. Well, actually it's from their mothers. Anyway, nude mice, 50% of them don't have T cells from their mothers, and they don't tend to have a higher incidence of tumors than normal mice. And so the idea that the immune system is constantly surveying, looking for tumors, kind of died. And then the rag mouse and the skid mouse were discovered, and they don't tend to have many more tumors than normal mice either. There's been a resurgence of the idea of immunosurveillance. And there is some evidence that if you take a T-cell deficient mouse and you give it like MCA under the skin, by themselves they don't have more tumors than normal mice, but if you give them tumor-inducing agents, they will get more tumors than normal mice. I don't think that's very good evidence for immunosurveillance, but there you go. It has recrudesced the field. So why do I think that tumors on the whole don't get rejected? So first, we no longer think that you get a tumor every 3 minutes. We think that tumorigenesis is rare because of genes like P53 and so on that monitor cells and tell them to die when they do bad things. And when a tumor does arrive, I don't think the immune system in general is going to respond to it. And the reason it's not in general going to respond to it is that tumors don't look dangerous. You know, what's wrong with a tumor? What's wrong with the tumor is that it's a cell that has lost growth control. So it's dividing when it shouldn't, right? It's making a bunch of cells. It's not looking dangerous, at least in the beginning. It's simply a cell that has lost growth control, right? It's dividing or multiplying. I remember the day that I discovered that division and multiplication in biology are the same. Yeah? And suppose this is a tumor that was expressing tumor antigens in MHC. Let's call them T antigens. And suppose you had a T cell that could see that. This is a viral antigen, a mutated antigen, an early embryonic antigen. What's going to happen to that T cell? Somebody tell me. It'll get signal 1. And?
Not signal 2.
And not signal 2. And? Die. Right? Signal 1. Go ahead.
Hi, Polly. Yes, I was thinking about it, but I also tried to relate that to
Wait. If you're going to talk for a long time, you have to go to the microphone. Right? If it's just a quick question I can repeat, then you can just say it. Yeah?
So I said I was thinking about the foreign antigen and relating that to the peripheral tolerance we talked about last week. So, and you just said like a few minutes ago that tumor will express high amount of self-antigen.
Can do.
Does the dose play a role, antigen dose, play a role in peripheral tolerance?
Good question.
So, because in that way they are still expressing the self-antigen, but some of those self-antigen will activate T cells, even though it's only signal 1. And most tumor will have the inhibitory co-stimulated, as we said. So how come some of the T cells become activated?
Okay, so that's more than one question, so let's do one first. All right, so first, does the dose of antigen matter to tolerance? And there are theoretical reasons for saying yes. So this was originally thought about by Mel Cohn, who I still think is the greatest immunological theoretician out there. And he was talking about tolerance in the... Okay, I want to start with a couple corrections. I've gone back and listened to the first 2 sessions and realized that, I make mistakes all the time. Some of them are trivial. So, for example, Brecher and Cohn's paper came out in 1969, not 1967. It was, of course, Archimedes and not Socrates who jumped out of the bath and ran around. And some of them are not so trivial. For example, in going through co-stimulators, I said that CD40 ligand was a strong co-stimulator of T cells. And that's actually incorrect. There is one paper which is missing a very important control, which suggests that CD40 ligand is necessary during T cell development and that it signals into T cells. But most of the evidence is that CD40 ligand is used by T cells to bind to CD40 to educate the cell that has CD40. So it's not so much that it's a strong co-stimulator by itself of T cells, it's that it's used by T cells to superactivate or license antigen-presenting cells. So in a way, that also makes them more co-stimulatory for T cells, but it's not the CD40 ligand itself. Right? So that was an important one to fix. Okay, so today is tumors. Last week we did fetuses and surgical transplants, right? And the conclusion there was that even though the fetus is foreign, it's not rejected because it doesn't look dangerous, right? At least if it's doing its normal development. And that surgical transplants are rejected because they look dangerous, because of the damage done by the surgery or the reperfusion injury, or even sometimes the disease that killed off the first graft. So if fetuses aren't rejected because they don't look dangerous and transplants are rejected because they do look dangerous and they're both foreign, why aren't tumors rejected? So we know that there are tumors that express foreign antigens, either virally encoded or mutated proteins or molecules that were up really early in fetal development, long before the immune system arrived, and then get turned off. So early fetal antigens. Or they sometimes express much higher quantities of thymus. And here's what he said. All right, so first you have T cells of varying affinity, or avidity, since they have more than one receptor. But let's talk about affinity for any particular antigen. At what avidity cutoff do you want to induce tolerance? And he argued that you want the same avidity cutoff that would induce activation in the periphery. If the avidity for tolerance is lower than the avidity necessary for peripheral activation, right, then you're going to tolerize T cells you don't need to tolerize. And T cell development is a pretty complicated thing. It takes energy to do that. So you don't want to kill off a whole bunch of cells that you don't need to kill off. Right? If the avidity for tolerance is higher than the avidity necessary for peripheral activation, then there will be a set of T cells that you have not tolerized that can be activated. So he argued that you want the same avidity for tolerance as for activation. Now, Charlie Janeway had a paper. By the way, before Charlie got into PRRs, he did a lot of experiments. He was a pretty sloppy experimenter. But he did have a paper showing that
Can you say what PRR means? Sorry.
pattern recognition receptor. Sorry, what is PRR? Pattern recognition receptor. Invented by Charlie Janeway. Brilliant. He had a paper showing that tolerance induction in the thymus might require a threshold about 1,000-fold lower than activation in the periphery. I don't know that that was ever repeated by somebody else. Maybe it was, and if somebody out there listening to the videocast knows about that, let me know. But theoretically, you would argue that it should be the same. All right? So, if you're tolerant in the thymus of a particular concentration of something, we're going to come back to that during autoimmunity, right? And you have a cell in the periphery that now expresses a lot more, you will have some lower avidity T cells that have escaped tolerance in the thymus that can now activate in the periphery. However, if that periphery is healthy, what's going to happen to them? They're going to get signal 1 without signal 2 and die. Now, signal 1 without signal 2 is a real simplification, right? We talked about Sinclair's idea and long before the discovery of all these co-inhibitory molecules, that there are co-inhibitory and co-stimulatory molecules, and they're not only expressed on APCs. Right? The co-inhibitory molecules are also expressed on heart and so on. So signal 1, I think Mel was wrong on that one. Signal 1 is a heads up that allows a cell to become sensitive to a whole bunch of co-stimulatory molecules and a whole bunch of co-inhibitory molecules expressed on cells. By the way, it was Sinclair's idea about this that finally allowed me to accept some data that I'd been having trouble with all my immunological career almost. And that is the fact that if signal 1 causes cells to die if they don't get co-stimulation, then how come you can stimulate cells in culture with anti-CD3? Is anybody worried about this besides me? So Sinclair's idea actually solved that for me. Because when you stimulate cells in culture, actually, let me tell you one thing Germain suggested. He said, signal 1 causes a cell to die, but you can overcome that with more signal 1. So if you give a lot of anti-CD3, you can make the cell independent of co-stimulation. Right? That doesn't work for me because it means that if you have a cell in the periphery that expresses a lot of an antigen that is not in the thymus, you ought to be able to activate it with only signal 1. Right? So that explanation doesn't work. But here's what did work, and that is when you put cells in culture, they're on plastic. And you stimulate them with anti-CD3, which is on plastic, right? Either Cephidex beads or on the plastic, right? There are no co-inhibitory molecules there. All those co-inhibitory molecules that are there on normal tissues are not there in that dish. So you have your heads-up signal, right? And you turn it on because you don't have the co-inhibitory molecules on the dish. But you do have co-inhibitory molecules on tissues. So it's that balance.
And you can address that experimentally by putting co-inhibitory molecules.
You could absolutely test that experimentally.
You actually don't get activation.
Has somebody done that? Hang on, let me repeat that so that the video people can hear. So you ought to be able to test that by putting co-inhibitory molecules on the dish. And that's been done?
I would have to look for papers.
You'd have to look for papers. Well, look for them.
Yes.
Okay, I keep asking people to send me those papers. And
Okay.
actually, Howard Young sent me one. I forgot to send it to you. I will send that to you.
The picture you showed there is of a solid tumor. Is the situation where the malignancy is in an antigen-presenting cell or a B cell any different?
So the question is, this is a solid tumor. Is the situation any different if the tumor were a B cell or an antigen-presenting cell? And the answer is yes and no. So there are, as far as I know, no tumors of functional dendritic cells. There are tumors in dogs called histiocytomas. And I think that they are Langerhans cell tumors. And there's two types. There's one in